So a multi-point confocal is a confocal with a camera. It's called a spinning disk as well, and we will see why. So we have again the same sample, and then we have the objective underneath. So it is an inverted system still, no light, and then we have lasers. So multi-point confocals work with lasers. And then the laser light reaches the dichroic mirror, and this is defocused this time. So it's not because it's not because we use lasers that it has to be focused. So defocused light, and the light reaches the spinning disk. So it's a disk. As you can see, there's a lot of little holes. So it's a metal disk, and it has lots of small holes in it that are organized in a concentrical way like this with a spiral. So that's what it looks like. And the disk is turning very, very fast. So I will show you a few movies so you can understand why is it that we do not see through, that we do not see the metal, basically, part of the disk. So the light that comes from the laser shines through all these little holes, through the objective, and it reaches the sample. And because the disk is turning very fast, we scan the sample. So this spinning disk is a scanner. It does scan like hundreds, thousands of points of light on the sample very, very quickly. So the sample starts fluorescing, and all these little dots of lights that are illuminated start sending their emitted light, and then the emitted light goes back through the disk. And because we have the focal plane of the objective and we have the focal plane of the disk, we have two conjugated planes, so it's a confocal. Okay, exactly in the same way as for a single point confocal. So this is also a confocal. And the emitted light goes back through the pinholes of the disk, and each pinhole is going to act exactly in the same way as the pinhole on the single point confocal. So it will eliminate the autofocus light by only allowing the main lobe of the point spread function to go through. And then the emitted light is going to reach the emission filter and then be scanned again on the whole camera. So on the whole camera chip, which records all these little dots of light. And then there's a digital image that is formed, and if we look at the digital image, we can see that we have pixels. It's made of pixels, and each pixel has an XY coordinate and one intensity. So uniform intensity per pixel. So spinning disk confocals are confocal. They are real confocals, and they are fast because they are camera-based systems. We illuminate many XY points at the same time, and we have many double cones of illumination in Z. So we do have an illumination in Z as well, but the light that comes from the autofocus plane is illuminated by the pinhole. So exactly in the same way as in a single point confocal. And the camera records both the XY coordinates and the intensity. Then we have the excitation light is scanned on the sample by the spinning disk, and also the emission light is also scanned onto the camera. So we get some sort of double scanning that is coordinated. And this can be maybe an image of a spinning disk that has a slightly different conformation. So sometimes on some spinning disks you can get double disk, and they rotate together and they have the same patterns. And in one of these disks, you have actually micro lenses, and these lenses focus the laser. So in this case, we have a focused illumination that goes through the objective. So many spots, focused double cones that go through the objective and are scanned on the sample, and then the emission is also focused and comes back and is sent to the camera. So we can have spinning disk systems with or without these micro lenses. And so either we have a cone of illumination, or we have either we have a cylinder of illumination, many cylinders of illumination, or we have many double cones, but the principle is exactly the same. So why is it that we do not see the holes when we image? I will show you through this little guy. basically the disk spins very fast compared to the acquisition time, so much, much faster than the camera can record. So for that reason, the for that reason, the camera sees through the disk, and you can see this little guy when it's turning, and we see all these lines there on the side. But if it goes fast enough, we do not see the lines anymore, and it looks like the circle is transparent, and maybe also some bad things happen to the poor little guy who's running there. But so this is the idea. It's the same idea as when someone on the bicycle passes by you. If the bicycle runs fast, you do not see the spokes on the wheel. It's the same way. So it looks like you can see through the wheel. That's exactly the same principle. So basically, this disk spins much faster than the camera images. So then we can see through. And so there are advantages. So what are the advantages of a single point confocal compared to a multi-point confocal? Because in a single point confocal, we have only one spot. So why do we image with only one spot if we can image with multi-point spots and also have an optical section? There are many advantages to single point confocals, and basically we have the flexibility of the scanned region. So we can decide completely flexibly how much of the sample we want to scan. On a camera-based system, the scan area is going to be a square or a rectangle, and that's it. And on a single point confocal, we can scan an area of any shape and any size all the way down to a single point. So we have a very high flexibility of the scan region, and that can be very useful. For example, I've shown you an image of a zebrafish with kaede, which when you shine light of a certain color on the green fish, some of the cells that are exposed to this light will become red, right? And so you want to shine light in a quite precise manner, not everywhere. So you want to make sure that you only activate kaede at these places. And this you can do very nicely on the single point confocal because you can decide exactly where you're going to image. So there's a high flexibility in the scan region, but there's also a high flexibility in illumination power. On the single point confocal, the laser light is concentrated into a very small spot. So we have a very high power there. Therefore, we have high bleaching and phototoxicity as well. But this is very useful when we want to, so we can modulate this illumination light very well. And it is very useful for techniques that require very high illumination power, for example, FRAP. Where we want the light to be concentrated into a small spot, and we bleach or photoactivate a small region of interest, and then we image the whole field of view. Or for STED, when we need to also have a very small spot of light to improve resolution. Oops, I've lost my mouse, here it is. We also have the flexibility of pinhole size. That is very important. So we have seen that normally the size of the pinhole should fit very nicely around the main lobe of the point spread function. And if we change wavelengths, or if we change numerical aperture of objective, so we will see all this tomorrow, when we change wavelengths or objective, the point spread function is going to change. And therefore, we would like to be able to open and close the pinhole in such a way that we have the perfect settings for this particular objective and wavelength. And on a single point confocal, it's possible because we have only one pinhole and it's an iris. We can open and close it. It is not possible on a spinning disk system because the pinholes are just a hole in a metal plate. So we have no choice. We cannot open them and close them. It is possible to change the whole disk, but that is quite, you have to physically go there and change it. So it's quite demanding. Whereas on a single point confocal, it's very easy to open and close the pinhole. So we have even more flexibility on a single point confocal when it comes to pinhole. And then we have the flexibility of a pixel size on a single point confocal. On a camera-based system, the pixel size depends on the size of the camera sensors. So the physical device is inside the camera. And that depends on the magnification as well. We're quite fixed. It's quite strict. And we will see all this again next week. But with a camera-based system, basically the pixel size, there's very little that we can do to change it. and we will see later why we need to change it. Whereas on the single point confocal, the pixel size is this distance between the scanning spot. So if you remember when I've shown you the single point confocal, we shine some light on one spot, and then we collect the emission light, and then we move to another spot. And when we excite that, and we collect. So we can choose how long we jump between the sampling spots. And that leads us to changing the pixel size. So we have a super high flexibility of changing the pixel size, and we will see next week how we change this and why. But basically, we can choose the perfect pixel size for any objective and any wavelength. So basically, on a single point confocal, we have a lot more flexibility. And we also have on a single point confocal the flexibility of the emission filters. So we can very often we can open and close the emission filters the way we want. We can set them to the range that we want. When we have spectral detection on the point confocal. So extremely useful to be able to adjust the emission filter to avoid bleed-through. So this is basically it. The single point confocals have a lot more flexibility than camera-based systems. But there are some tricks that can help both the camera-based system and the single point confocal, because the single point confocals, we said they are quite slow, right? So the difference is number one, first difference is the speed. So the camera-based systems are much faster, but there are some tricks that can help a single point confocal go faster. And these are here. You might recognize them when you image. With your single point confocal, you can choose bidirectional scanning. And so you will have to find also where the bidirectional scanning setting is. All single point confocal have that. And that allows you to go faster. We will see this also next week. We can use a resonance scanner. So the normal scanner on the single point confocal is called the galvanometric scanner. But you may have a second scanner that goes very fast, and it's called the resonance scanner. We clearly have several detectors, and that allows us to go faster. And if we have a spectral detector, we can also image several colors at the same time and go faster. Yes, Juan?
In regards of the to how it was the bi-directional scanning, that will be for
Bidirectional
next week.
scanning.
That will be for next week, right?
Yes.
Okay, yeah. So I can say my question for that time. Okay.
It's all right, that's all right. But you know about this bidirectional scanning, you have used it maybe.
Yeah, I have used it, but there's an issue that maybe it's because the it's not completely aligned the when scanning, but I see some kind of lines.
Yes, I understand. So
Yeah.
bidirectional scanning, you need to realign the two lines basically.
Mm-hmm.
And there are some ways where you can do this automatically, or you can help manually. Sometimes it's not perfect, but most of the time it does a very good job. In general, if you image at a really high resolution, the bidirectional scanning, you might see some small shift. And this is the reason why if you image at high resolution, very often you will use the single directional scanning. But if you image like a slightly less like a slightly lower resolution,
Mm-hmm.
then the bidirectional scanning is fantastic because the tiny shift, even when it's aligned, sometimes there's a tiny shift, and this you cannot see when you have a lower resolution. So there are some limitations to the bidirectional scanning as well, but it does
Okay.
help you go faster,
Okay. Thanks.
double the speed basically. And we will talk about this next week. The second difference between the single point and the multi-point confocals is the flexibility. So I've told you the single point confocals are a lot more flexible, but we have some tricks that can help a little bit the systems that have a camera, so the multi-point confocals. So we have, for example, we can change the field of view, the size of the field of view by cropping the camera. So that is one thing that we can do. And then we can change a little bit the pixel size because on some microscopes we have an additional lens that we can put into the light path that will change the pixel size. We can also make the pixels bigger by binning. So there are a few tricks that we can do for changing the pixel size, but it's not at all as flexible as the single point confocal. And when it comes to the pinhole size, yeah, sometimes we can get an additional disk, but it's the disks that we get are anyway only optimal for one objective and one wavelength. So even if you have two disks, they are not going to be perfect for all the different wavelengths that you use and all the different objectives. Whereas this is the case on a single point confocal, you can change the pinhole size to be perfect for any objective and wavelength. And then when it comes to the emission filters, some microscopes have several cameras, or sometimes you can use one camera chip to image several colors, and you get a much smaller field of view. And this we will see also next week in the lecture about several colors. But it is, in general, it becomes quite expensive, and you have to align them. So it's still quite rare that you would have several cameras on a system, unfortunately. So in general, you're stuck to the physical filters you have. So we will see all this in the speed versus noise workshop, and also where we will compare single point confocal and multi-point confocal, and also in imaging multiple colors at once next week. So the conclusion here is that when you use a single point confocal, the great advantage of a single point confocal is the flexibility of setting everything perfectly. So if you use a single point confocal without adjusting any of the flexible parameters, you would gain a lot in using multi-point confocal. So I think that there are not spinning disks everywhere, but I mean, our facility has 3 spinning disks, and I think it is important to know what the advantages are of a single point confocal. If you use a single point confocal, you're going to spend more time by the microscope because it's a slow system. And I think it is important that you use them in a correct way. Okay, so don't just pick randomly the scan size and the number of pixels that you put in there and the objective. But hopefully after this course, you will know what you're doing. That's the idea. Right, so now I will talk a little bit about light sheet, and I just want to check how I'm doing this time. Yeah, I think I'm okay. So light sheet systems are systems that have been invented a very long time ago, but because of technical developments, let's say, both on the camera side and also on the sample side, so on the clearing of the sample side, there has been a big revival of light sheet systems, and they are now a big player on the market. So I do not think that anyone in this course is using a light sheet. Normally we have one person per year. A light sheet system may look like a box like this. It may also look like that. And so there's a very the strong differences, let's say, in the design of the light sheet systems, but they have some things in common. And so you can have different architectures, but the principle is that the excitation and the emission light path are perpendicular. So you have seen that in all the systems that we have considered so far, when we image fluorescence, both the excitation light and the emission light go through the objective, and there's only one objective. Okay, so we have excitation. The excitation light reaches the sample fluoresces all over the place. Some of the emitted light will go back through the objective and go to the sensor. But on the light sheet system, it's different because we have 2 objectives in general. So again, there's different tricks, but and I will show you one trick that Leica has been using before with a single objective. But in any case, even if you use a single objective, the excitation light and the emission light needs to have a perpendicular light path at the sample. And that means all sorts of different things. It means that we have optical sectionings and only the plane of interest is illuminated. So when we image in wide field, the whole field is illuminated, included in Z. So the whole volume is illuminated like a big cylinder of illumination, and we image the whole thing. So we focus on one plane, but the light that comes from above and below, because above and below the sample is also illuminated, it also fluoresces. So the fluorescence that comes from above and below the focal plane will also participate in the image. This is wide field. On a single point confocal or a multi-point confocal, we have spots of illumination, but it's either lots of cylinders or spots in inverted double cones. So also the fluorophores above and below the focal plane are also illuminated and they fluoresce. But because we have pinholes, we throw away the light that comes from above and below the focal plane, but we do excite them. We do photo damage, right? Above and below the focal plane. We just throw away the emission by using a pinhole. In a light sheet system, and in a multiphoton system, it's different, but it's sort of the same principle that we only illuminate what we image. So we do not excite above and below the focal plane. And therefore, we also have an optical sectioning. So this is what our light sheet looks like. And you can see the two objectives, the excitation objective and the illumination objective. And then here we would have our sample, and we would dip the two objectives in the liquid that covers the sample. And then behind, so the light is going to come from here, and then the sample fluoresces, and the emitted light that goes into this objective will be sent to the two cameras. In our case, we have two cameras. Okay, so there are many different types of architecture, but the principle is the same, is at the sample, we have a perpendicular light path for the excitation and the emission. So this is yet another example for Zeiss. So in Zeiss, they would have the Zeiss light sheet. They have also two objectives. This is the excitation objective. This is the illumination objective, the emission objective. And here we would put the sample in the middle in some sort of tube, so it hangs in some agarose cylinder. And you can see here the light, which is shaped like a sheet. That's why it's called light sheet. So we will see a little animation afterwards that will show you how this sheet is formed. But basically, we have this sheet of light that only illuminates like a knife, you can say. It sort of cuts the sample, and it only illuminates the plane of interest, and only that plane is fluoresces, and that light goes into the emission objective, and it's imaged. Okay, so we always have light sheet systems are always camera-based, so it means they're very fast, but they have low flexibility. We always have a perpendicular excitation and emission light path at the sample, and only the plane of interest is illuminated, okay? And we have no emission from the other planes, and because of that, we have no photo bleaching, no photo damage from the other planes. And though we have an optical sectioning, we only illuminate the plane of interest. So we have a high Z contrast compared to wide field. But in the emission light path, it's exactly the same as wide field, which means that this object, all the light that comes from the illuminated plane will go into the emission objective, and then we have an emission filter, and then we have a camera. So it's exactly the same as a wide field system in the emission light path. Okay, so here is the little animation from the Leica system that I think they have stopped now, but this was quite interesting because it shows how the sheet is formed. In this light sheet system, they actually use only one objective, but with a little trick with mirrors that you will see, you anyway have the excitation and the emission light are perpendicular to each other at the objective. So let's have a look in here. I think it's quite well done. This is what I like here. Here we see the specimen. It's probably Drosophila. And here we have actually on top of the condenser. This is the traditional light sheet where we have two objectives, and we would have the sheet of light that sort of cuts the sample. But in their system, they have a little mirror device like this. They have some device that they put at the top, and that allows them to use only one objective. So the sample goes in between. Here we have one objective. And they're sending the light on the mirrors. And this is, so they send the light to the mirror. And what I like is that they're showing you how the sheet is done. So you can see how they scan the light very quickly until it goes so fast that it actually looks like a sheet of light, even though it's a beam of light that is just scanned very, very quickly. So now if we look at the sample in the middle, we get the sheet of light that is there, and then we simply move the sample through the sheet of light and we can image it exactly as the same light path as emission light path as a wide field system. Okay, so it's a quite nice little video that explains how they make the sheet. And that is the way all systems, all light sheet systems make the sheet. They're basically a beam that they scan very, very quickly. So the challenges with light sheet microscopy, it sounds like the perfect world, right? But there are some challenges. Making the sample transparent, it's not easy, so we will talk about clearing later. We need to mount the sample between all these objectives. There's not much space, so sometimes it's difficult to mount the sample. And also any imperfection in the sample may create some shadows because the light comes from the side. But then there are different tricks from different companies. You can move the sample, you can move the sheet, etc. You can use an airy beam like in ours. So there are some tricks to get rid of shadows. And then you can, if it's a very big volume, then you have to maybe fuse the different volumes if you rotate the sample. And this is also creates some additional challenges in the processing of the images. And then in many type of light sheets, you have either a small field of view or a good Z resolution, but not both together. But on the airy beam, you can get both of them. So there are some tricks around this sort of challenges. And you can deconvolve also the sample, but in general, we image very big samples. And if it's not completely homogeneous, it also creates some difficulties there. But in general, it sort of works. The clearing and the matching refractive index are absolutely crucial in light sheet microscopy. And this is also because we have very big samples for that. So we can image like a whole brain, for example. So this is a little summary of what we have for the three types of systems that I have presented that all have an optical sectioning capacity. So we have good optical sectioning everywhere. The excitation and emission light path for the confocals go through the same objective, whereas on the light sheet they're perpendicular. On a single point confocal, we have detectors, and the other two systems are camera-based. And in here, the speed is very different for a single point confocal compared to camera-based systems. And the photon efficiency is super high for a light sheet because we do not use any pinhole. So any photon that is emitted is going to be collected. And we have anyway very low photo bleaching because we only with a light sheet because we only illuminate the plane of interest, and that is very different from a single point confocal where the light is concentrated and we have high bleaching and phototoxicity. And on a single point confocal, we have excellent flexibility, and that's not the case for the others. And we have flexibility also. I mean, we have flexibility also in the Z everywhere, but it's a little bit less in the light sheet because we have to reduce the field of view. And we have super high flexibility, basically, for a single point confocal everywhere, and less for the light sheet, the camera-based systems. And we can image at depth in semi-transparent samples. It is better on light sheet, but anyway, it's always better on all systems if the sample is transparent by clearing. And then all these light paths that we've talked about, after the workshop today, we will release a little summary so that you can have a look at them. So this is just when do we use which system? So again, a wide field system, the two confocals and a light sheet. If you have many large samples, wide fields are very good, spinning discs are very good, camera-based system very fast. So if you have lots of samples or they're very big in XY, like large sections, these are the first choice, definitely. If your sample is a little bit thick, then the spinning disc or the single point confocal are best because then you have an optical section. If you have a flat monolayer, you can be fine with any of those, actually, even with some light sheet systems that can take monolayer of cells. But if you have a large sample, light sheet is definitely like a thick sample that is transparent or clear light sheets are definitely the choice system. And if you have very fast events and dim samples, it's actually really good to have a wide field system because these are very fast and very photon efficient. Same thing for live samples. We can image basically live samples everywhere, but single point confocals are not very good for live samples because of the concentration of light, so it's high light toxicity. And if we have lots of overlapping colors, we'd want a single point confocal to have a spectral detector. And if it's very dim, both the light sheet and the wide field systems are really good because they're very photon efficient. So we always ask ourselves when we have a new project, can we use a wide field system because it's so efficient and so fast? If not, then we'd go for a spinning disk confocal. If not, we would go for a single point confocal. And if not, we go for a light sheet. This is a little bit our own workflow. Okay. So we will see all this in this afternoon, and we will look at the 4 light paths for each of the microscopes, and you're going to become super duper expert with all this. So I want to, I'm going to stop now, and we're going to go for lunch. But